Hello, how are you today? I hope you're wonderful. I got the Amoeba Music shirt on, which I should have wore when I had that Amoeba slide a couple slideshows ago, but I can't put two and two together so fast sometimes. So here we're at Mendel stuff. This is really the simple Mendel stuff, and then a little bit more complicated like sex-linked stuff we'll get to in the next set of slides. So here we go. Looking again very closely at the syllabus. Here's explain the inheritance of genes and traits as described by Mendel's laws. Very general. That's the what do they call that thing that show? I forget what they call that thing. But that's the objective, right? The learning objective. That's what they call it. So here we have the idea of the sets of crosses that Mendel did. They're not going to ask you this, but you kind of can explain what they're going to ask you through these. So he had these pure or true breeding ones that he called the parental generation. None of that language is in the syllabus, right? Creating hybrids, crosses of two different kinds, and then producing this generation that he called the F1. So here's the basic idea in terms of the results and things that you will get asked, right? So when he crossed those first ones, and I'm going to use P's for no pun intended, kind of, for gene symbols, right? These were the parental ones. There were again, homozygous, we would call that, pure, right? Dominant, recessive. When he crossed those, all the ones he got had the same phenotype as that. That's the one he called dominant. That was his first basic idea, right? And then these F1s, which are now right here, the ones that had two different kinds of genes for this trait, when he crossed two of them with each other, right, then he got these noteworthy 3-to-1 ratio results that the rules of probability would say you would get if you have certain rules involved in this system that you have. And it's those rules that he came up with and called the law of segregation, right? So when you cross these, right, we'll now make a simple Punnett square. Remember, this parent represented here as two different genes. When meiosis happens, they separate. So when you write them a little separated from one another, you are simulating meiosis happening. This other parent with the same two different genes is going to also produce two different kinds of gametes, and therefore you get kids that have this dominant purple gene in the ratio of 3-to-1, right? 3-to-1 phenotype. Look at this, right? Not in the blue box, but here's where they tell you students need to calculate genotype and phenotype ratios, and often it's a common student error that they confuse those. So if they ask you for a genotype ratio from this F1 cross, you would say one of these homozygous dominants to every two of these heterozygous to every one of these. But if it's a phenotype ratio, they ask you it's three purple ones, because purple is a phenotype, for every one white one. White's a phenotype, three dominant for every one recessive. So this is the rule, right? What's his law of segregation say? Oh, look at, they want you to know that. Law of segregation and independent assortment we'll get to next. How can that be applied to genes that are on different chromosomes? The linked genes are coming in the next PowerPoint show. So here's the highlights of his ideas about how this works. Those ideas he called the law of segregation. Each organism has two factors slash genes, right? One they got from mom, one they got from dad. When each organism has babies, those two genes are going to separate. Segregates the word that he used. During what? During meiosis, when gametes form. So the gametes are just going to get one from each pair. That way, when two gametes come together, that offspring that results is going to get one from mom and one from dad, one from each pair from each parent. Now, going to chromosome land, like Mendel never could, right? We know these genes are segments on chromosomes, and it's because homologous chromosomes segregate, separate, when in meiosis 1, remember they wanted you to know that we get this phenomenon that results in the F2 generation, that 3-to-1 ratio. So now we have the idea that, again, it's this math that told Mendel this is what's going on. And in the next unit, we're going to look at the idea, the simple idea that if you don't get the math you expect, and your expectations are based on Mendel's laws like segregation, then that means you have something else going on. Something like linkage, something like sex linkage, whatever, whatever. Okay? So here we have in the syllabus here, the pattern of inheritance, monohybrid, dihybrid. That's not really a pattern of inheritance. This is not worded well. Sex-linked and genetically linked, that's next unit, but those are patterns of inheritance. Monohybrid simply means I'm only going to look at one trait when I cross these things. I'm only going to look at two traits. I'm going to call that dihybrid, right? But organisms have thousands of traits. It depends on how many you look at in terms of whether you call it this or this. And so I don't know how they would work that into a question. Yeah, there you go. More former students calling from like 100 years ago. Sorry. Here we go. When then he took two traits and looked at two traits and did the same set of crosses, I got a homozygous dominant parent, homozygous recessive parent, parental generation. I crossed them to get the F1s, right? They are heterozygous, but now for two traits. When I cross them, what do I get? I get this 9 to 3 to 3 to 1 ratio of phenotypes. 9 that have both dominant phenotypes for every 3 that have one dominant, one recessive. We've gone over this a million times, right? Why does this work? He said, how does this work? It appears to me that the only way I'll get these numbers in this F2 generation when I do this set of crosses is if each of these two traits is independent of each other when they segregate the things that cause these traits and assort themselves, one from each pair, into new little gametes and then therefore new little babies. This is what I will get only if they don't have anything to do with each other. And if you do this kind of cross again and you don't get 9 to 3 to 3 to 1 phenotype ratio, that means they're probably linked, right? That means something else non-Mendelian's going on. Okay, so now these are not in there except for this. Explain deviations from Mendel's model. They specify sex-linked and linked. They don't specify other deviations, and this is not much of a deviation. And this example here still follows the law of segregation idea. So they could describe this to you and say, you know what? When you get two of these kind of genes in you that are different, you don't have one dominate the other. You get this in between color. If they explain the rules to you, they would still be able to expect you to do, okay, let's say you had one pink parent. I'm going to use P's again, that we've already told you is a result of heterozygous, and then one parent is white flowered, and we said that's homozygous. Can you tell me what kind of kids they would have? And you would say yes, half, right? Genotype wise, half and half, but also phenotype wise, in this case, half pink, half white. They could ask you that. Okay. Then you have the very similar idea that if you have two genes that don't dominate each other, but you see both phenotypes, we call that codominant. But since that language never comes up in the learning objectives in the blue boxes, they won't expect you to know like that term or whatever, but they could tell you the story. And then also the idea that sometimes there are more, in fact, more often than not, there are more than two kinds of genes. Here again in blood type, we have three kinds of genes. And if they tell you a story about three kinds of genes, but then they also tell you that even though there's three different kinds, you only have two of them, like maybe this two, and they separate when you make gametes and da-da-da, then they expect you to be able to apply the law of segregation to this particular phenomenon. Okay, so then they do have this. Many traits are the product of multiple genes. I think what they mean here is polygenic inheritance, but they don't use that term, which they really should because they used to. But that's all I can think that this one would mean. And then again, they explain the rules to you. And we still have the idea so far that for each of those different kinds of genes, multiple genes, you still only got two of them, but in this case, all these genes go to determine just one trait, right? That's the idea of polygenic inheritance, right? So these traits often show a distribution that is across a continuum, like from light to dark, which when you map the frequency is often in this bell curve thing. I think they might throw that, I don't know, because they don't have that word polygenic in the syllabus. So here we have the basic idea that the environment also plays a part in what phenotypes end up. Like if you stay out in the sun longer, that's going to darken your skin no matter what kind of genes you have, yada, yada. Physiological processes, I guess a trait is a product of that. I guess this might be what they're talking about. We'll get to another example later also. So here they say that patterns of inheritance, we might tell you a story. We might give you data, like these two parents had this many kids of this kind, or we might give you a pedigree. So they expect you to be able to take a pedigree like this and say, like, whether or not this trait is dominant or this trait is recessive. To remember the pattern you look for to answer that question, is the trait dominant or recessive? You look for two parents that have the same trait, in this case, the colored in one. If they have even one kid that has a different trait than they do, then that kid must have the recessive trait, because two recessive parents could never have a dominant kid. It couldn't be the other way around. So that's the trait you always look at. Here, to contrast, these two parents have the same trait. It's the not shaded in one, and they have a kid that is shown shaded in. That means in this one, the shaded in trait is the recessive one. This one, the shaded in trait was the dominant one. And again, that also tells you that these two parents must have been heterozygous. That would be a very common question. And therefore, any kid they had would have the possibility of getting a recessive gene, even though neither one of them shows it. And so there you go with pedigree stuff. So that's, so you gotta focus in on that to make this make any sense, and then it makes all the sense in the world. And it's so funny that I think I'll end on this one. So next time, just one left for this unit, non-Mendelian stuff. Have a great day.